DNA's ability to self-assemble in a predictable manner offers a novel route to manufacturing tailored nanomaterials. Complementary DNA strands hybridize due to base pair interactions, and by programming these interactions, numerous strands can be combined to build complex nanostructures. One commonly used method is DNA origami, where a long DNA strand, called a scaffold, is folded into a predetermined geometrical shape by numerous shorter strands known as staples. This method has been used to produce both 2- and 3-dimensional structures with a variety of functionalities. However, every structure made with the DNA origami method requires a custom set of staples, which can be costly. The DNA brick method offers an alternative route to creating 3D nanostructures. The method permits a large variety of different structures using the same library of DNA strands. These DNA strands are 32 nucleotide long bricks, allowing for 4 connections to adjacent bricks, connecting similar to Lego building blocks. Unlike DNA origami, similar structures use mostly the same collection of bricks with only a few custom strands, which reduces costs and saves resources. However, DNA bricks have fewer connections between helices than DNA origami, and this may potentially have impacts on the mechanical properties of DNA bricks. We simulated 2 128 base pair long rods, one built using the DNA origami method and the other the DNA brick method. The structures were simulated for 165 nanoseconds, and both of them adopted a slight twist as expected. The average distance between DNA helices was found to be higher for DNA brick structures, resulting in a slightly higher cross-sectional area for the DNA brick rod. The larger helical separation was found to originate from the structure of the junctions, which in DNA bricks contain fewer connections than in DNA origami. DNA brick had a higher separation than DNA origami at its junctions, where the connections should be their closest, meaning its junctions had a weaker binding strength. The dihedral angle plots indicate that DNA bricks have right-handed junctions and DNA origami have left-handed junctions. The DNA brick structure, due to its fewer connections at the junctions, was observed to have a slightly lower persistence length in our simulation, and was overall more flexible than the DNA origami structure. Despite their compact structure, DNA origami plates are permeable to ions. Next, we investigated how the DNA brick method affects ionic permeability. A pair of 2-helix thick plates were built using the DNA origami and DNA brick methods, and a bias was applied normal to the plates. The DNA brick plate had a higher rate of ionic flow, or ionic conductivity, than the origami plate. The origami plate blocked ionic current more because of its tighter packed helices and smaller cross-sectional area. Overall, DNA brick structures are found to be more flexible and have a larger cross-sectional area than DNA origami structures. To automate design of DNA brick structures, we developed LegoGen, an online toolset for converting 3D models into DNA brick structures for both simulation and experiment. You can access this toolset at nanohub.org/tools/LegoGen, or by looking it up on the web.